All right, so let's go ahead and get into blood and the blood vessels, which is really one of the main things we're focusing on this week. So we're going to talk about blood, the different parts like plasma versus serum, formed elements, and talk about all them. We're going to talk about the different blood vessels and how blood flows through, as well as get into like how do we get blood from the capillaries back up to the heart and blood reservoirs, as well as some disorders. So let's go ahead and talk about blood. Blood is a connective tissue. It's a little bit weird compared to other connective tissues because the matrix in this case is a fluid. It's a liquid, which is different compared to most other connective tissues where there is some sort of gel-like or solid matrix, right? So this is a little weird. The normal pH is about 7.4. The range for the pH is very narrow. It's like a 7.35 to a 7.45 really. It's a real narrow range for pH that's considered normal. And the average adult has about 5 liters of blood. The viscosity is a little bit thicker than water. So it's watery, right? It's not like it's like a maple syrup kind of thing, but it's a bit more viscous than water. I'm not going to make you memorize like the osmolality and all that, but we do want to talk about what we find in the blood, and that's we have a liquid matrix and then we have cells that are suspended in that liquid. The liquid part is what we call plasma, and that makes up over half, about 55% of any blood sample, any given blood sample, and that the solid parts are about 45%. And the solid parts are the cell parts, so erythrocytes or red blood cells, leukocytes or white blood cells, and thrombocytes or platelets. We'll talk about each of those types of cells and what they do in just a little bit. But let's talk about plasma first. Plasma is going to be the extracellular fluid. It's the fluid outside of the cells that we find inside the blood vessels. When we look at the plasma, right, so remember plasma is just over half of any given amount of blood. Most of that plasma is water, right, 92%. And that makes sense when we think about how liquidy, how fluid blood is. And then mixed in that water, we have a handful of other things. We have electrolytes, which make up a fraction of a percent, things like sodium, potassium, calcium, phosphate, bicarbonate, right? These are all ions that we have there. And then we also have proteins that make up a good chunk, about 8%. So these are proteins that are typically made in the liver and then get pushed into the blood. We have things like albumins. Albumins are the most common, and albumin's main job is to help retain fluid in the blood, right? So having this high concentration of albumins is going to help keep the water with it, right? Basically draws water in. And so this way we can keep the electrolytes, we can keep the water so that our blood volume and therefore blood pressure does not drop. And then we have globulins. We're going to mainly be talking about what we call immunoglobulins, which are antibodies, right? And we'll talk about the different classes. We also have clotting factors, so like fibrinogen. Those are going to be things that are used to help clot the blood if there is an injury or some sort of damage that has occurred to a blood vessel. There's a bunch of different clotting factors. We're not going to get super deep in the weeds with that. And then the other little things like hormones, right, that are going to be, those will fluctuate depending on what the body's doing, right? What age are we? What part of the cycle are we in? If we're talking about reproductive ones like estrogen or whatever, like any sort of hormones that are moving around can fluctuate depending on what's going on. Now, plasma is that's how we describe the matrix inside the bloodstream. Serum is similar, except we've removed all the clotting factors. So we've had some coagulation happen. Sometimes labs require plasma, sometimes they require serum samples. And so this is why sometimes, like if you go and you have to have blood drawn, they use multiple tubes for your samples, right? Why you're like, why do they have to take like 3 sticks of blood or whatever? It's not just because they need all of that blood. It's also because inside of those tubes, there are different compounds to either allow things to clot or not clot, to do something to stabilize the blood in a way for specific tests, right? And so that's why we make that distinction between plasma and serum. So if we're looking at, let's say we're just looking at blood cell count, red blood cells, white blood cells, platelets, we want plasma collection. And so we have to add anticoagulants into the collection tube. So heparin, citrate, things that are going to prevent blood clotting. Because if the blood clots, it's irreversible. We can't do anything with that. So again, some things we may not want that. We may not care if it clots, or maybe we do need it to clot, and so we won't have those anticoagulants. But again, that's why sometimes when you get your blood drawn, you may have multiple sticks taken, right, because the collection tube is going to have specific compounds in it that will put the blood into a specific state so we can make like certain assessments or measurements. Now, plasma volume is regulated to maintain blood pressure. We have osmoreceptors in the hypothalamus that are going to release ADH, right? If we lose too much fluid, that way we can retain more water in the kidneys, so we concentrate the urine more and don't allow as much water to escape, right? We have a handful of things that we can do to help maintain things like blood pressure, but we'll get into blood pressure a little bit later. Let's go ahead though and get into the formed elements now. Again, these are the cellular components of the blood. We have a basic hematopoietic stem cell. This is your generic multipotent stem cell that has the ability to become several different types of blood cells. It can become a white blood cell. It can become for platelets, it can become red blood cells. And through a series of steps, through a series of exposures to different compounds like interleukins, we can mature into different types of blood cells. So if we're looking here, we have our hematopoietic stem cell. If we're exposed to like IL-7, we're going to go down the lymphatic, or the lymphocyte route, I should say. If we are going down here for IL-3 or GM-CSF, right, we can become other types of white blood cells like monocytes, or we can become the granular white blood cells, or we can become something like a red blood cell or platelets, right? So this is a good thing to kind of look at, just to kind of have an understanding of how we start with a single generic stem cell and how different signals are used to allow for us to differentiate into all the different blood cells we have. Now, this whole process is happening in the bone marrow. So the bone marrow is responsible for making all of these different blood cells and allowing most of these maturation processes to occur within the bone marrow as well. Hematopoiesis, which is the process of this blood cell development, this has to happen regularly because blood cells and platelets have very limited lifespans. Some of them can live for years, but others only last for days. So like the standard red blood cell has an average lifespan about 120 days. And so we're constantly making new blood cells to replace those that are lost. So let's talk about red blood cells first. Red blood cells, also called erythrocytes, their main job is really just to transport blood gases, namely oxygen. They do transport other things like carbon dioxide as well. However, oxygen is their main player, and this is because they have this pigment called hemoglobin that these blood gases can bind to. The hemoglobin is also what gives us the red coloration because it contains iron. So similar how like an iron oxide or rust is kind of orange red, that's also why the blood is colored red from these guys. They have this kind of donut-like shape, what we call a biconcave disc. But if we're talking casually, it's kind of like a donut to where the hole did not go all the way through. They are anucleate, meaning they do not have a nucleus. And they also lack mitochondria. Part of the maturation process for an erythrocyte is to basically boot the nucleus and the mitochondria and organelles and whatnot out. So they're kind of empty, and that's to make as much room as possible for hemoglobin, because the more hemoglobin we can pack on these guys, the more oxygen and everything we can transport around. As I mentioned, their lifespan is relatively short. We're looking at about 4 months, all right, where they circulate around the bloodstream. They are relatively, they're small as far as cells go, and their immature ones are a little bit bigger. We call those reticulocytes, and so they'll still have some of those components that haven't got booted yet. Platelets are even smaller. They're very, very small. These are actually not entire cells. They're fragments of cells called megakaryocytes. So megakaryocytes in the bone marrow will actually pinch pieces of themselves off. And so the platelets are those pieces that have been pinched off. Because they're not an entire cell, they also do not have a nucleus. They're very, very tiny. And we have tons of these, right? Tons and tons of these. We're looking at thousands of them per microliter of blood. Their lifespan, though, is very, very short. We're talking in the days range, right? Even shorter than the red blood cell. Their whole purpose, though, is for blood clotting. And so they're going to be able to kind of trigger that blood clotting and be part of that process. We'll get into that a little bit later. Now we get into the white blood cells, of which we have several categories. So we're going to go through each of these categories. We're going to start with the lymphocytes and the monocytes, which are the agranular white blood cells, meaning they do not have a grainy appearance. The lymphocytes have a few different types: B, T, and NK cells. B named for the bone marrow that they're mature in, T for the thymus, and NK stands for natural killer. So these are going to be cells that help with broad, like, immune functions. They're really specialized in this. B cells primarily are antibody producing. T cells are going to provide cell mediated immunity, and NK cells are going to be, well, as the name implies, natural killers. These guys are going to have like a large round nucleus. We don't really have the granules, which is why we're considered an agranular white blood cell. And they make up a good chunk. You know, we're looking at 20, 30, 40, 50% of white blood cells, depending, right, on whether or not we've caught anything lately. They're also pretty small. The monocytes are going to be phagocytes, meaning they're going to engulf things like bacteria, fungal cells, debris, right? They're going to engulf and destroy things that way. They have a large kidney bean shaped nucleus, so it's like that fat U shape. They do have granules, but they're really small, so we still kind of call these guys agranular. And they're going to be a much smaller percentage of white blood cells. We're looking at under 10%. They are much larger, though, in comparison, size-wise. Then we get into the granular white blood cells. We have the neutrophils, the eosinophils, and the basophils. Neutrophils are kind of your first responders. They're going to be the ones who show up kind of right at the beginning if we have an injury or an infection. And their whole job is to go in and just phagocytize, just engulf bacteria and fungal cells that they find. Their nuclei are usually multi-lobed, so it almost looks like a string of beads, from 2 to 5 of these lobes. The granules tend to be a bit pink when they stain, and this is going to make up most of our white blood cells, right? So over half, for the most part, are going to be neutrophils. They're large, and one of the things that they do form is pus, right? Pus is actually white blood cell material. It's not something from the pathogen. It's a response driven by your own body. Eosinophils, they have a little bilobed nucleus, so we see 2 lobes. Their granules tend to stain red. They make up a very small percentage. We're looking at, a couple percentage points, so 1 to 6%. They're a bit larger, but the big thing eosinophils do is they are cytotoxic against parasites. Usually when we think eosinophils, we're thinking worm parasites. So, some sort of invading worm is what we relate eosinophils to. They also will help downplay things like inflammation. So they can kind of counteract the function of our next white blood cell, which is the basophil. The basophil is going to release things like histamines and is going to increase things like inflammation as well as our responses to allergens. And so we do see a link between basophilic activity and allergies. that these guys are more active during an allergy. Their nucleus is bilobed and they stain purple, like a deep purple. They are the rarest of the white blood cells. We're looking at less than a single percent of white blood cells are going to be basophils. So now we've gotten through all the different blood cells. Let's go ahead and talk about the different blood vessels, which they all have the same basic structure. And I'm going to go through some of this pretty quickly because we have a lot of this information down already. Veins and venules are going to carry blood to the heart, right? Arteries and arterioles are going to carry it away from the heart, and capillaries are going to be our drive-throughs. They're going to be the waste and nutrient and gas exchange. So you remember that color does not tell us what blood vessel we're looking at. It can give us a hint, but we do not use color or oxygenation as our defining tool. We use direction of flow in relation to the heart to define blood vessels. So if the blood is moving from the heart away, it is artery or arterial. If it is moving back towards the heart, it is a vein or venule. And if it's really small in these big net-like messes, then it's a capillary. So arteries, again, carry away, and they have a few key structural components that allow them to do that. One is elasticity. That blood is being pushed through arteries at a high pressure because the heart is actively pumping it and pushing it. And that means arteries have to be able to withstand that high pressure. And elasticity is going to allow for some of that stretching, right, every time the heart beats. Contractility is we have smooth muscle inside of it that allows the arteries to either open, right, vasodilate, or kind of pinch inward or vasoconstrict. We have different types of arteries. We have elastic arteries, muscular arteries, and anastomosing arteries as well. So we see like elastic, we see is close to the heart. Muscular, we see we branching off. And this arteriovenous anastomosis, right, we're seeing that transition. So elastic, let's get in some details. They are the largest in diameter, so things like the aorta, right, would be part of that. They have a lot of elastic fibers. They don't have as much smooth muscle because they're not going to be as involved with blood pressure. Their job is to just move blood and withstand the high pressure. So during systole, when blood is being actively pumped by the ventricles, the walls can distend because they're elastic. They can stretch to accommodate that pressure. And then when the ventricles relax, we can recoil, forcing blood forward. So we kind of snap back to normal size, and that pushes blood forward. During diastole, there's a little bit of pressure, right? And that's going to help just maintain blood pressure and flow. As we go from elastic arteries, we move to muscular arteries. We're getting a little smaller in terms of the inside diameter. There's more smooth muscle and less elasticity. And so these are the ones that are going to undergo things like vasoconstriction and dilation to help control blood flow. They're going to give us a little bit of control over blood pressure with the muscular arteries. So if we look at what's going on with the elastic arteries, right, we see that the elastic ones can stretch, right, when blood is actively being pushed through them. And that as blood continues through, we see that we snap back in, and that is going to push blood forward. And then lastly, we get the anastomosing arteries. These are where we see things like arteries branching together with the same, supplying the same region. So we look at things like in the hand and the feet, right? We see these archways, like the different palmar arches. These are going to give us different routes to get to the same tissues or organ. If they don't have this, then it's considered an end artery. Connecting from the arteries is going to be the arterioles, which are really small. We're almost a capillary, but not quite. So we're getting smaller, we're closer to the size of a capillary instead, but these guys are going to play a very big role in controlling and regulating blood flow from the arteries. They're actually going to have a good role in terms of maintaining blood pressure. So similar to the muscular arteries, they will go through vasoconstriction and dilation to help regulate blood flow. We then get to the capillaries. The capillaries are super teeny tiny, like 1 cell thick type blood vessels. It's just one single layer, simple squamous epithelium, what we call endothelium when we're looking at capillaries, and a basement membrane wrapped around. Because they're so thin, it's really easy for us to make exchanges, right? For nutrients and waste products to swap, as well as gases like oxygen and carbon dioxide. So we can make those swaps because they're really thin. You don't have to get through a lot of cells to go from the bloodstream to the tissues or from the tissues to the bloodstream. Now, there are different types of capillaries depending on where we are in the body. Most of the time, we're talking about continuous capillaries where the cells, the endothelial cells are close together. We find these kind of in your fatty tissues, in your central nervous system, in your muscles, right? And so most things are not going to slip through there. When we look at capillaries involved with the kidneys, the intestines, and endocrine glands, where bigger substances kind of need to be able to squeeze through, right? Like waste products that contain nitrogen. If we're looking at the kidneys, the intestines, we have to bring in things like amino acids from the food we eat, endocrine glands that secrete large molecules like hormones. We have things that are a little bit bigger that need to slip through, and so because of that, we need slightly bigger gaps or holes. And so we're going to use fenestrated capillaries in that case. And so we look at the picture, we see like there's all these little pores, these little openings that these bigger items can pass through. And then when we're looking at things where we need entire cells to potentially get through, that's where, like if we're looking at the bone marrow or the liver or the spleen, where we have really big things that have to get between, in and out of the bloodstream, this is where we look at what we call discontinuous or sinusoid capillaries, because again, the reason we have these differences is because what we need to get through, right? Continuous is going to be the most narrow. It's going to be the strictest in terms of what can pass through in and out of the capillaries. Fenestrated, we're allowing certain bigger things to be able to pass through in terms of molecules. And then discontinuous or sinusoid, we want big enough holes where entire cells are able to get in and out. We continue talking about the capillaries, right? We form these capillary networks. So it's not just a single capillary branching from the arterial side to the venous side. We have these almost like net-like structures, right? But these nets, right, are going to be controlled. We have what we call precapillary sphincters that are going to help control how blood flows through these capillary beds. These precapillary sphincters are found on the arterioles. They can open or close so that we can shunt blood into certain directions. That's kind of what we're aiming at with those sphincters. If the sphincters are relaxed and open, we can flow through that entire net. If we close off some of those, we can ensure that we are pushing blood, in a direction away from where they're shunted off. And as we get through the capillaries, we make it to the other side, to the venous side, where we have the venules and the veins. Venules are kind of like the veins side of the arterioles. They are little tiny vessels coming from the capillaries that are going to merge with the veins, and their whole job is to take blood from the capillaries and put it back into the main bloodstream, which is the venous side. Veins are going to continue from venules. We don't have as much elastic or smooth muscle tissues because we're not under the same pressure that the arteries were, so we don't need them, and so our walls are thinner compared to an artery. And we have the addition of the veins have an addition of valves to prevent blood from flowing backwards, because we don't have that pressure pushing blood forward. We need something to prevent blood from draining back down. If the valves get weak, this is where you end up with things like varicose veins. We do have some vasodilation and constriction, but it's not the same as the arteries, really. And the veins, actually, most of your blood is, if you were to guess where a single, blood cell was, your best guess would be to say a vein, because most of the blood volume we have is found in a vein at any given moment. So, we mentioned they don't have a lot of smooth muscle, they don't have, a lot of elastic fibers and what, they're kind of flimsy, and so how the heck do we make blood go from the feet back up to the heart? There are a couple things. One, valves, which we mentioned, but we also have what's called the respiratory pump and the skeletal muscle pump. The valves, again, just are one-way doors that blood passes through, and then it cannot pass backwards. But the respiratory pump is actually using your respiratory system to help with pumping blood. Because when you breathe in and out, you actually have changes in pressure in your thoracic cavity. And when the pressure in your thoracic cavity goes down, right, like when you breathe in, your blood actually will be kind of pulled upward. And when you exhale, the pressure increases again, right? And so we're using that change in pressure of, every time we breathe in, the pressure dropping and kind of drawing blood up from the abdominal cavity upward into the thoracic. That's going to basically keep blood flowing forward, right, back towards the heart. Skeletal muscle is going to work as a pump, because when the muscle is is contracting, that's going to put pressure on veins. A lot of veins run between muscle groups, and so it's almost like if you were squeezing something with your hand, how if you were to squeeze something with your hand, you think a cow in their udder, squeezing on the teats of the udder to get milk out, right? The skeletal muscle is kind of doing the same thing. It has that same squeezing action to try to basically force, squish blood up through the veins back up towards the heart. And the valves keep the blood from flowing backwards. So the skeletal muscle squeezes on the vein, that pushes blood up towards the heart. And then when the muscle relaxes, the valves snap shut so the blood can't drain back down. Now, when we're at rest, where do we find the blood typically, right? Most of the blood is, over half, is going to be found in a venule or a vein. Some of it will be in the pulmonary vessels, in the lungs and whatnot. Some will be in the heart. A bigger chunk will be in the arteries and arterioles, and some will be in the capillaries, but most is going to be found in the veins and venules. Now, what can go wrong? The blood vessels can experience physical changes like a stretching or balloon-like swelling. This is called an aneurysm, where the blood vessels walls are basically not holding out. They're not able to push back, and so they balloon outward under pressure. Sometimes this can lead to what we call dissection, where the aneurysm rips open, and this leads to internal bleeding. There's a lot of different causes. Sometimes you're born with it. Sometimes it's things like atherosclerosis or high blood pressure, diabetes, that can increase your risk or help you develop those. Most of the time we're looking at these in the aorta, like a thoracic aorta or an abdominal aorta, aortic aneurysm, although they can occur anywhere else as well. Now, varicose veins we mentioned earlier. This is when usually we think about this in the legs, but the thing is the valves are not properly closing. The valves have been damaged, and so because they cannot close, you have blood flowing back downwards. We do see this happens a lot in people who have to stand for long periods of time, people who are obese, older people, pregnancy, because the developing fetus puts more pressure on abdominal veins. These are all contributing factors. If you have to stand a lot, the idea of moving your legs, so walking, things like that can help. Using compression stockings and elevating the legs can help as well. This is why sometimes too, when people go, if they have to fly a lot, they'll wear compression stockings or compression socks when they fly, to help with this, right? So anytime you have to stand or sit for a long period of time, this can be problematic. There are treatments including sclerotherapy, laser therapy, ligation, and stripping, where it's kind of cool to watch, where they go in, they inject something in there, and they seemingly disappear like instantaneously. It's kind of cool. Atherosclerosis, this is a disease in the arteries. Basically, fatty plaque has been deposited into the blood vessel's walls, and because the plaque is being deposited in the walls, rather than the vessel growing outward, the plaque builds up more towards the inside, so the inside of the vessel narrows, and that constricts blood flow. This can be because there's damage to a blood vessel. This can be because of bad cholesterol, like a high LDL cholesterol, which is the bad cholesterol when you hear about that. Sometimes these plaques can develop into a clot. And the things that increase your risk are things like smoking, having high blood pressure, diabetes, high cholesterol levels, and that this is a lot of deaths are due to atherosclerosis because someone ends up having a heart attack or a stroke on top of having this. Another thing that can occur is deep vein thrombosis. This is a blood clot that forms in a deep vein. Usually we're talking about in the legs. This causes pain, swelling. This also can lead to what we call a thromboembolism. Basically, the clot has dislodged itself and has now flowed through the cardiovascular system and then ends up in the lungs, where it causes a pulmonary embolism. So this is something that can be deadly. This is also why when you have surgery, or like if you fly a lot, if you do anything that where you don't have a lot of room to move, or you haven't been able to move for a while, this is why they want you to get up and walk around. So after you have surgery, they want you up and moving so that you don't develop this. Again, it's also why people may use compression stockings. Or, like, let's say someone's had surgery and they're not going to be very mobile. They may have devices that compress and squeeze on the legs, right? So to kind of stimulate or simulate the moving that someone would do, if for whatever reason the surgery doesn't allow them to get up and walk around soon. Sometimes we do need to give medications that will break down clots or that prevent clotting in the first place, just in case. This is also a rare complication of certain birth control pills and birth control, like hormonal birth controls that are out there, because it does increase your risk of clots with that. That being said, right, that's a conversation to have with a doctor in terms of whether or not you have other risk factors that could increase your risk of DVT if you were to use something like a birth control.